In America, 1 in every 4 deaths is attributed to a form of heart disease, and regenerative therapy can be used in the future to treat these patients. Previous studies have involved transplanting undifferentiated embryonic stem cells and induced pluripotent stem cells, which then differentiate into cardiomyocytes. However, this procedure has a high risk of teratoma formation.
Toyama and colleagues utilized the glucose metabolism of cardiomyocytes to purify them from ESC and iPSC. Glucose is converted into pyruvate through glycolysis, and pyruvate enters the TCA cycle as acetyl-CoA. They wanted to figure out the difference in metabolism between cardiomyocytes and undifferentiated stem cells. They first did a transcriptome analysis on ESCs and cardiomyocytes purified via the mitochondrial method. They found that genes involved in glycolysis are upregulated in ESC, while in cardiomyocytes, these genes have lower expression. On the other hand, the genes involved in TCA cycle have higher expression levels in cardiomyocytes than in ESC. They also did a fluxome analysis where they incorporated carbon-13 into glucose, which is then metabolized by cardiomyocytes and ESCs. The results show that most of the carbon-13 in cardiomyocytes end up in TCA intermediates, while in ESC, carbon-13 level is higher in glycolysis metabolites. In addition, they found high level of carbon-13 ending up as lactate in ESC, which is mostly discarded. These findings led them to hypothesize that maybe cardiomyocytes is more effective in using lactate as its metabolic source than ESCs. They grew ESCs, non-cardiomyocytes, and cardiomyocytes in media containing lactate and no glucose. They found that within 96 hours, all cells died except cardiomyocytes. So now they confirmed that cardiomyocytes can survive in lactate supplemented media without glucose, while other cells cannot. They call this method the Lactate Method. They also tested ESC and iPSC's embryoid bodies at different lactate concentrations for 7 days, and they found that 4 millimolar lactate supplemented media with no glucose optimally selects for beating cells.
After finding the concentration of the lactate solution, researchers wanted to find the ideal period of differentiation. In other words, what period of time will give you the highest reproducibility, purity, and yield of cardiomyocytes? They found that this period was about 20 to 30 days of incubation. How did they do this? Well, the researchers used an EDU incorporation assay, which is an alternative non-radioactive assay that uses 5-ethanol-20-deoxyuridine, or EDU for short. This method uses fluorescent molecules attached to a DNA precursor that is incorporated in solution during S phase. With this, they could measure the proliferative activity of cells. The results showed that after 30 days of EDU incorporated cells decreased, and they implied that cells must be sensitive to the lactate method. In a separate experiment, they measured mRNA expression of cardiomyocytes-related genes and non-cardiomyocyte-related genes, and what they found was that cardiomyocyte genes had increased, while pluripotency-related genes and non-cardiomyocyte genes decreased. Can you cut that out? The researchers also tested for the percentage of alpha-actin before and after metabolic selection to test the efficiency of lactate method. Because alpha-actin is expressed during cardiomyocyte differentiation, researchers used differentiating cells like human ESC cells and iPS cells to test the alpha-actin concentration for the lactate method. The results showed that all cells had higher alpha-actin percentages after metabolic selection and a higher number of cardiomyocyte cells present after differentiation. In conclusion, researchers determined that lactate method, which had a yield-based efficiency of 74.4%, was way better producing cardiomyocytes than the former mitochondrial method, which had a yield of 52.9%. So why do cardiomyocytes survive better in the lactate method? To figure this out, researchers used carbon-14 to measure the lactate uptake activity in neonatal and human ESC-derived cardiomyocytes. And what the researchers found was that cardiomyocytes had higher rates of activity compared to other cells. And when they measured ATP levels, they found that cardiomyocytes sustained longer levels of ATP than non-cardiomyocytes, meaning that cardiomyocytes could effectively take up lactate and make ATP efficiently, helping them live longer than other cells.
While on the subject of cardiomyocytes' high uptake of lactate and how they use it for metabolic energy production, the scientists wanted to look into how lactate was transported by something called the non-carboxylic acid transporter, or MCT. These MCTs are abundantly stored in muscle cells and tend to localize in plasma and within the mitochondria. To test its function, the cells are treated with the MCT1 inhibitor under glucose-free and lactate-rich conditions. With the MCT inhibitor in place, these cells' survival were drastically reduced. This helped to establish that the lactate uptake through MCTs allowed the cells to survive longer under metabolic conditions. After this knowledge, expression of MCTs in cardiomyocytes and HESCs were found to have very little difference. However, after electron microscopy on the HESCs and cardiomyocytes were taken, it was found that the cardiomyocytes had substantially higher amounts of mitochondria. Due to these passive transporters, it is assumed that the lactate uptake in cardiomyocytes may be so effective due to the concentration gradients created by the active TCA cycle. The proliferative capacity of purified mouse and human ESC-derived cardiomyocytes were then tested by assessing the rate of cytokinesis. The purified cells were dissociated and placed sparsely on meth-layer dishes and then counted 4 times over 8 days. The purified cardiomyocytes proliferated up to 2.5 times over the 8 days. Using glass microelectrodes, the purified cardiomyocytes demonstrated nodal, atrial, and ventricle-like action potentials. They also were able to respond accordingly to increased and decreased beating frequency depending on the dose, along with responses to temperature and calcium concentrations. This shows that these purified cells will act and respond just as they would normally in the heart. Finally, human ESCs, non-purified and purified cardiomyocytes were transplanted into the testes of non-obese diabetic mice. 2 months later, 90% of the ESC mice had tumors, 40% of the non-purified cardiomyocytes had tumors, and 0% of the purified mice had tumors. As mentioned in lecture and demonstrated by these results, if cardiomyocytes are not completely purified from ESCs, microteratomas can develop from pluripotent ESCs.
In conclusion, the researchers found that the lactate method was more effective in purifying cardiomyocytes than the old method, and has a lower risk of developing cancer. Theoretically, a patient needs about 10 to the 9th power purified cardiomyocytes before it can be used for therapeutic application. In this study, the researchers obtained 5.3 times 10 to the 5th power purified human cardiomyocytes per dish. Doing the math, we can calculate that 800 dishes are necessary to obtain the required amount. In the future, this can suggest that combining a more sophisticated differentiation of the aforementioned lactate method could be a realistic application for pluripotent derived cardiomyocytes for human therapy.